distinguished professors, honored guests, Professor Emeritus Patricia Gaspar, dear colleagues and friends. It is my great pleasure to welcome you all today to this special conference dedicated in the memory of Professor Miskolczy Dezső, an exceptional scholar, neuroscientist and intellectual whose work continues to inspire generations of researchers and doctors across Europe. Today, we gather not only to reflect on the achievement of a remarkable scientist, but also to celebrate the values that defined his life: intellectual courage, academic excellence, humanity, and a profound commitment to education and research. Miskolczy Dezső belonged to a generation of scholars who understood that science is not only the pursuit of knowledge, but also a responsibility towards society. As a pioneering neurologist and psychiatrist, he contributed significantly to the development of modern neuroscience and psychiatric medicine in Romania and Hungary. His teaching and his institutional leadership left a lasting mark on our university. For our university, the legacy of Professor Miskolczy Dezső holds a particularly significance. His name is inseparably linked to the development of medical education and scientific culture in Târgu Mureș, at a time when the foundations of modern higher medical education in this region were being consolidated. Through his academic leadership and his dedication to training future generations of physicians, he contributed decisively, shaping the intellectual identity of this university. For our institution, remembering his work is not only an act of historical respect, but also a reaffirmation of the values upon which our academic community continues to build today: professionalism, integrity, innovation, and science to society. During some of the most difficult and turbulent decades of the 20th century, he defended the dignity of academic life and the autonomy of this university. At a time when political pressure often threatened intellectual freedom, he remained committed to ethical principles and scholarly integrity. It is therefore especially fitting that this conference also gives us the opportunity to welcome one of today's leading neuroscientists, Professor Patricia Gaspar from Sorbonne University, Professor Emeritus at Paris Brain Institute, French National Center for Scientific Research, whose research on serotonin and brain development has profoundly influenced neuroscience. Her presence among us creates a symbolic bridge between the pioneering spirit represented by Professor Miskolczy Dezső and the discoveries shaping neuroscience today. We are deeply honored that she has accepted our invitation and will share her expertise and insights with us during this conference. On behalf of our rector, Mr. Leonardo Zanfire, and myself, I would like to express my sincere gratitude to Professor Gaspar and to the organizers to making this conference possible.
Let me introduce now briefly and present Professor Miskolczy Dezső's life. Miskolczy Dezső was born in 1894 in Baja, Hungary, and he obtained a medical degree from the Budapest Royal Hungarian University in 1919. He started his neurohistological research under the mentorship of Karol Schaffer, a world-renowned neurologist and brain researcher. You probably remember the Schaffer collaterals in the hippocampus bear his name. And a major change in his life was probably the Rockefeller Scholarship in Madrid, where he worked with the Nobel Prize winner Ramon y Cajal. And they had a very good friendship, so they remained corresponding friends, and he also, so Professor Dezső had a very good feeling for languages, so he translated even some works of Cajal to German language, so they remained friends until Cajal's death. And he shared Cajal's motto in minimis perfectio, so perfection even in the smallest things. So throughout his life, Miskolczi's life, we can follow this main idea. In 1930, he was nominated professor at the university in Szeged, where he organized a very modern clinic, also with facilities for neurosurgery and neuroradiological investigation. So these pneumo and ventriculography were the first imaging techniques used for this purpose, and they were really standing for about half a century later until the MRI and the new techniques occurred. He also established a brain research institute there with a biochemistry and histological research facility. You will see that everywhere where he established the university, he also followed this research laboratory. So he started to develop, to found these research laboratories. In 1940, the medical university moved back from Szeged to Kolozsvár, Cluj, where he was appointed as director of the neurological and psychiatric clinic. And in 44, in the university year, 44-45, he was the rector of the university. So he had a key role in maintaining the continuity of medical education at this university. In the same year, the Hungarian language University of Romania was founded and later became the Bolyai University in 1945. And the medical faculty of this university was moved to Marosvásárhely, Târgu-Mureș, our city, so we can consider him as one of the founders of our university. Here he became director of the neurological and psychiatric clinic, and here we can see where they started their work. So these very poorly equipped psychiatric observation, probably you know we today call the Lenin Street Hospital, but later in 1950 he already made the plans, and according to these plans a new building was renovated where the neuropsychiatric clinic was moved, and here he also established a neurohistology laboratory. As a recognition of his successful research work, the Romanian Academy of Sciences established a scientific research center, is you know the building near our main building in the campus. And he became the leader of this research center. He was a devoted teacher. Here we see among his students, he was beloved by his students. His lectures were followed by many students, and he was a devoted leader of the hospital. So here we see him as the director of the neurology clinic in 1964. He returned to Hungary after near a quarter of a century in Transylvania, and in Budapest he was appointed as the head of the Department of Neurology in Budapest at the Institute of Postgraduate Medical Education, and he held this position until the age of 75, and he passed away at age 45 in 1978 at Budapest. If we look at his scientific achievements, his studies covered almost entire fields of neurohistology and neuropathology. At the beginning of the 20th century, there was the debate of neuron doctrine, so it was not clear where we have a continuous reticular structure or distinguished isolated neurons communicating with each other. Cajal and also Miskolczi supported the neuronal doctrine, which later proved to be the right one. And he published also with Károly Schaffer the influential book, The Histopathology of the Neurons, and he refined Bieszowski's silver impregnation, which also was used by Cajal in his works. So the very nice drawings of Cajal partially are using these silver impregnation method. He worked a lot on the heredodegenerative diseases, and his main idea was that both a genetic background is required, but also some external conditions, activating external conditions are necessary for these illnesses to occur in clinical stages. He worked on the lipoidosis, on the Niemann-Pick, Tay-Sachs-Schaffer and Hans-Schuller-Christian syndromes, and he worked very much here in Târgu Mureș on the mosquito-related encephalitis. It was an epidemic in Transylvania, so he could prove that the pathological changes were similar to those in panencephalitis. So these results attracted great international attention. And it's significant that at Cluj he organized the fourth dedicated neurosurgery department in Europe, and later this kind of hospital was established also here in Târgu Mureș. He published many clinical observations, and he was also interested in the organic foundations or morphological foundation of the mental disorders. And his work, the histopathology of schizophrenia, or anatomical basis for the symptoms of schizophrenia, or initial symptoms of mental disorders, are still cited today. So even some Oxford scholars are citing and recognizing his very important role in establishing these morphological background of these psychiatric diseases. He also worked in the field of endocrinological diseases, just a few titles like the growth disorders, diseases of the pituitary gland, or the hypothalamus pathology. He learned here, he was originally a Hungarian citizen, but here he learned Romanian and published in Hungarian, German, and Romanian languages. Just a few examples. It's this paper in Romanian language. He published also papers on the history of medicine. You see a paper about Gheorghe Marinescu. He wrote a textbook for medical students that was the biggest textbook at that time in the 1950s. And he became a member, he was elected a regular member of the Hungarian Academy of Sciences in 1948. And later, he became also a regular member of the Romanian Science Academy. So actually, he's the only person who I know that was in the same time full member of both the Hungarian and Romanian scientific academies. So I would say that he was a bridge between the Romanian and Hungarian scientific communities. So I can consider that Miskolczy Dezső was one of the greatest professors of our university, and in 2012, on the recommendation of Professor Péter Somogyi from Oxford, our university and the regional committee in Cluj of the Hungarian Academy of Sciences jointly established this Memorial Lecture. So each year we propose to invite a world-leading scientist to honor his legacy. And I would like just to show the last part, who were the invited lecturers until now. The first lecturer was Professor Péter Somogyi himself from the University of Oxford, followed by Professor Vizi Szilveszter, who was the president of the Hungarian Academy of Sciences, László Csiba from the University of Debrecen, Freund Tamás from Budapest, also president of the Hungarian Academy of Sciences. Later, István Módi from Los Angeles, Thomas Klausberger from the University of Vienna. And this is just a picture from Thomas Klausberger's presentation. This is the same room where we are now. It's the old style, so when it was just a festive hall of our university, so it was also used for teaching purposes sometimes. And after that, during the epidemics, we had two online award lectures by Professor László Acsádi and Berecki Daniel from Budapest. And after that, we started in 2024 with Professor László Vécsei from Szeged, and the last lecture was Professor Tamás Dóci from the University of Pécs.
Professor Patricia Gaspar is Emeritus Research Director at the Institut National de la Santé et la Recherche Médicale, INSERM, and the Institute of Biology Paris-Seine, Sorbonne University. She has had a long and distinguished career in the field of neuroscience and neurofarmacology. After an initial training as a neurologist and neuropathologist, she opted for a career in research. She obtained her PhD degree in molecular pharmacology at the Paris University, and in the early years she was visiting professor at Vanderbilt University in the United States of America. She started studying the anatomy of the catecholaminergic system in humans and their alterations in Parkinson's disease. In the US, she worked on primate cortical anatomy. After returning to France, switched to neurodevelopment and started her own research group in Paris, trying to map how brain circuits can be modeled by the environment during early life and how this could influence vulnerability to brain disease. In parallel, she focused on the developmental role of serotonin and its implications in brain circuit modeling. Using molecular and genetic approaches in mice, her team demonstrated the developmental role of serotonin on neural circuit maturation, sensor system underlying behavioral changes. Analyzing molecular pathways, the group uncovered the transient developmental expression of serotonin receptors and transporters that are involved in these effects. Besides, the research group characterized the mechanisms underlying the diversity of 5-hydroxytryptamine Ray-Phe neurons and their implication in parental behavior and early life stress. Synthesizing these, Patricia Gaspar's research helped to link serotonin signaling with activity-independent mechanisms that refine brain circuits and brought mechanistic insights into the developmental basis of some neuropsychiatric disorders. Professor Gaspar has authored over 150 peer-reviewed publications with more than 17,600 citations and a Hirsch index of 65, reflecting her substantial impact on the field of neuroscience. She published in Neuron, Nature Communications, the Journal of Neuroscience, and other prestigious scientific journals. She is a member of numerous international scientific boards. She trained more than 20 PhD students and postdoctoral researchers. She was a grant reviewer for many organizations like ANR, INSERM, Wellcome Trust, Israel Science Foundation, Fonds National Suisse, Fonds National de Belgique, DFG, European Commission. She featured as invited speaker at scientific events in France, Europe, Japan, and America, and kept the Sackler Award Lecture in 1999, the Alfred Fezard Lecture in 2013, being awarded with the Camille Bollinger Prize in 2012. Throughout her career, Patricia Gaspar has consistently demonstrated scientific excellence, high-level creativity, and research quality.
I think the reason he invited me was because he noticed that my father was born here, and actually I have a history of being in Târgu-Mureș, because this is my grandmother in 1923, and this is the Avut family sitting in some place in Marosvásárhely, which was at the time the name of Târgu-Mureș. And this is my father on this lovely white horse with someone who looks really we can see it's another century that has passed here. And so this is, and so I'll just briefly tell you a little bit about my father, because he had an interesting trajectory that led him through many different countries and different also work experiences. So he, as I said, was born in Romania, which in Marosvásárhely, and he was actually mobilized in 1943 to go to the Russian front, where quickly after the war he was made prisoner in Germany, and after the war was then made prisoner in France, which was, I guess, lucky, as they was afforded the possibility to stay in France, and where he did his medical studies for a while. And then, because he couldn't really exert in France because of his the fact that he was born in Romania, he left for the abroad and worked during his career in Palestine, in Bethlehem and in Jerusalem, which was in the Transjordanian part. And then moved back to Tunisia, where he ended his career as a surgeon. And in the same time as being a surgeon, he wrote a lot of poetry for which he's quite acknowledged. And I think there's been some lecture about his life, I think last year, and I think somebody was organizing this last year. So this is about my father, and now it's about me. Well, I didn't take the poetry path nor the neurosurgery path, but I started being, as a medical doctor, and then shortly after became very interested in the development and in serotonin. So why serotonin? I guess most of you know about serotonin as an antidepressant. Many people are taking fluoxetine as an antidepressant. We know that chocolate is also a good, source of tryptophan and could help us in difficult moods. But we know that serotonin has an effect on multiple effects in biology. And I think there's hardly any physiological process that doesn't imply serotonin, whether it's blood clotting, bone mineralization, pancreas secretion, and glucose metabolism, and you name it, vasoconstriction and so on. And part of this is, and so shows that this system is not only in the brain, it's everywhere in the body. And it's also it's a neurotransmitter monoamine, which is very conserved in evolution. The first organisms to use this are actually unicellular flagella, who use serotonin already as a transmitter and as a signaling molecule. And the structure of the molecule hasn't changed much. The metabolic pathway also for the synthesis is almost the same. It's food, tryptophan coming from the food, which is then transformed to tryptophan hydroxylase by an enzyme called tryptophan hydroxylase. There are 2 isoforms, 1 and 2 in mammals. And then that is stored in vesicles with a transporter which is called vesicular monoamine transporter, or VMAT, and then released. And then for the clearance of this, so that the buildup is not too high, there is a transporter which takes back the serotonin into the cell and removes it from the synaptic cleft or whatever tissue where it is. And then it's degraded by different enzymes, including an enzyme called on mitochondria, which is called monoamine oxidase, and it comes in 2 isoforms, monoamine oxidase A and B. Then when it's released, serotonin can act on multiple receptors. There are almost 15 different receptor subtypes. They come in 7 families, main families, and 14 different subtypes. So the system is often discouraging to people because you have to deal with so many signaling mechanisms, and it's a bit, I had a hard time to understand all these receptors when I get into the field, and I still don't know much about all of them. So as I mentioned, so in the mammals and in the human and in preclinical models, you have to remember that serotonin is produced in the brain, but it's only 5% of the serotonin, which is produced by the raphe neurons in the brain. Most of the serotonin is produced in the gut by enterochromaffin cells, and there's some production in the vessels, in the thyroids, in the pancreas, in the bone, in the pineal gland, and the hematopoietic system. So these are local productions that are mostly doing locally an action. And recently it's been discovered that it's also produced in the placenta before the serotonin neurons mature in the embryo. Okay, so what about the brain? So we're going to concentrate on the brain. And so as I mentioned, again here, serotonin is employed in many, many, many different brain physiological mechanisms: appetite, arousal, sleep, thermoregulation, breathing, mood, anxiety, aggressivity, learning, you name it. But my interest was in development. And I started to be that essentially because I was in the lab of a pupil of Ramon y Cajal, Constantino Sotelo, who was doing lovely work on the neural development in the cerebellum. And I thought that was really very interesting to understand how the neural circuits were building in the brain. And there was some ideas that serotonin may be implied in these mechanisms. And this was based on some studies that's been in the 80s and the early 90s showing that in mollusks, in invertebrates like Elizoma, 5-HT could have an effect on the axon growth, either promoting growth or retracting the axons. A Russian scientist named Busnikov had shown that in sea urchins, serotonin is important in the blastula formation and in the neurogenesis and the production of neurons. And an American researcher called Gene Lauder had shown that the serotonin also is expressed in neural crest cells, and it's very important in the migration of these neural crest cells to different organs. So there was some evidence that there was some role there, and that was, that could be something interesting. So I started with a project and this idea that maybe serotonin and other amines, if they act during development, could have maybe, and having all these effects on development, could have some later effects. And this is justified by the fact that, as you can see here, that in the mouse, the serotonin raphe system develops very early from E10, and almost by birth, it's already almost grown to the whole body in the spinal cord. So it's a very early maturation. In humans, we know it's the gestational week 5, where the serotonin system starts to mature and starts to have an influence. And as I mentioned, has some effects on many different developmental processes. So my idea was that maybe this could influence something in the brain and the construction of the brain, and have some effects in maybe there was some notion about autism or psychiatric disorders that could be influenced by these developmental changes. So when I embarked on this, actually we started. What I'm going to show you is actually. I hope I'm not going to bore you too much, but I'm going to show you some of the work that I did on preclinical models in mice, mainly, trying to look at the consequences of changing the level of serotonin, either by increasing it, by reducing the clearance of serotonin with the genetic or pharmacological models, or decreasing the levels of serotonin with the genetics, and show you the consequences. So I'll go through many years and studies and try to summarize this. And I don't know if you can ask questions, but maybe in the end you can ask questions if there's something which is unclear. So let's start with and I was lucky actually to start with some models which immediately showed us and which are the effects of increasing brain serotonin. And these models are based on the fact that, as I mentioned, so when serotonin is released, as I mentioned, so it's taken up by serotonin transporter. So if you have a knockout of the serotonin transporter, or if you block it with fluoxetine, you're going to have an increase of serotonin, which is going to build up, okay? Another way is the monoamine oxidase. If that enzyme is reduced either pharmacologically with inhibitors of monoamine oxidase or with a genetic manner, then you're going to have a buildup of this. And at the time, a very important paper came out in Science from Hans Brunner, and they had shown a very interesting medical case. They had followed a case of patients who were not really patients, but they had mild mental retardation, but what was more characteristic was that they had an aggressive behavior, and there was a history in the family of arson, rape, and violent behaviors. So they were interested, and many of these men were in jail. And there was a genotyping that was done, and they discovered a point mutation of the monoamine oxidase A gene. And they showed that this was really linked to the aggressive behavior in these men. And at the same time, some colleagues in France, in Orsay, who are actually immunologists, had done a mouse model. In fact, they were working on a completely different story. They were working on interferon. They were putting a transgene to increase the level of interferon expression, but apparently that was not very successful. But they noticed that in one of their mouse lines there was an aggressive behavior. And because of the paper of Hans Brunner had come out at almost the same year, they thought maybe this could be the MAOA gene. And they looked at the gene, and they noticed that they could find that the transgene had been inserted in this gene and caused a loss of function of the MAOA gene. And when I met this Olivier Caz, he was measuring the level of serotonin in the brains of these mice. And if you look here at the level of serotonin from postnatal day 1 to the adulthood, you can see that these knockouts had a huge increase of serotonin during early life, and this was normalized later on. And the reason it's normalized is because the MOAB, there's another enzyme which steps in after, which normalizes the level of serotonin. So we started working on this, and on this mouse model, and I suggested to Isabelle Seff and to Olivier Caz to look at the barrel cortex. And the reason, because I had seen this paper of D'Amato and collaborators, where they had seen that in the mouse cortex and in the rat, there was a transient innervation, increase of serotonin innervation in the barrel cortex. I don't know if you know about this, but this is the somatosensory, primary somatosensory areas, and this would be this region here in the mouse cortex. And this is a flattened view of the mouse cortex that's been flattened and just stained with immunocytochemistry for serotonin. And what you can very nicely appreciate here with the serotonin is that there is very strong labeling of this primary somatosensory area, the visual area, and the auditory area. So I thought that could be an interesting model. So I convinced them that we should have a look and that we could look at that. And because this is a nice model where, just to resume, so the barrel cortex is the representation of the whiskers on the snout, so that one whisker is mapped exactly on one area of the cortex. So there is a one-to-one projection between one whisker and the brain through different relays. And this is due to each barrel here, the sensory afferents from one barrel converge into layer 4 of the somatosensory cortex, make these nice clusters, and the cells around them of layer 4 arrange around them, and the neurons in layer 4 make these very nice arrangements that you can see here with the Golgi staining oriented towards the center of the barrel. This is a very nice model to work on because it's easy to look at, and its topographic representation is very stereotyped. So when we looked at that, what we were lucky was to see that immediately that the barrel cortex here, that you can see here with a Nissl staining or with a CO staining in the normal mouse, in the knockout mouse, you cannot see this organization. It's a bit difficult to see, but you don't see this clustering. And which is more appreciating that you can see here is the lack of barrels on the flattened cortex. And it took us some time then to understand, to work out what was happening. And what we could see is that instead that the thalamic axons was making these nice little bouquets with the neurons clustered around them, they were actually, the axons of the thalamus were more broad. They had more extension. They had less branches, but these branches were more extensive. And the axons around, the neurons around them were not surrounding them properly. So this was found also later on with the serotonin transporter knockout and with after fluoxetine exposure. So it was due to the increase of serotonin. And what we could see is that this corresponds to a very specific time during development, because initially in the mice at P1, the thalamic axons have a very broad distribution. If you look at them, they would be like this. The single axons would be not so much branched and more diffuse, and make this staining pattern, which would be uniform. And as in a few days, these axons were going to mature and are going to make these nice little bouquets with these branching, and they're going to organize. So this is something which is happening over a period of 5 days after a life in the mouse. And so we could see that during this time, so if you increase serotonin during this time, you prevent this mechanism. And what we could see by genetics, by crossing these MAOA mice with the 5-HT1B knockouts, was this was due to this was almost entirely due to an overactivation of the 5-HT1B receptor, because in the if you cross the MAOA mice with a 5-HT1B knockout, you had a complete reversal of the phenotype, which became completely normal. And so what is this 5-HT1B receptor doing? So it's one of the 14 receptors, okay? But this one is expressed in the thalamic axons during development. It's a presynaptic receptor. So what it does actually is to, when it's activated, it controls the glutamate release. So when it's activated, if you activate it too much, you inhibit the glutamate release. So you inhibit neural activity, okay? And also if you overactivate it, another way is that you decrease cyclic AMP, and cyclic AMP in turn has some effect interfering with axon guidance, so that some of the signals of axon guidance, such as ephrin A, B signaling, I don't know how much you know about these, but it's molecules which cause the retraction of growth cones. So if you interfere with this, the guidance of the axon will be modified, okay? So this is for the 5-HT1B receptor, so which can have an effect both reducing the activity and interfering with axon guidance. So this effect, interestingly, this effect has a very, very short critical period, meaning that if you treat the animals with an inhibitor of monoamine oxidase or with fluoxetine from P0 to P7, you perturb the development of the barrel cortex. If you do this before or after, you have no effect. So this is something that's really acting at a very short time. So this induced us to wonder whether this might explain some of the effects, the paradoxical effects of antidepressants that have been reported in the literature. So we know, you all know as clinicians maybe, or clinicians among you, that if you give SSRIs, fluoxetine, you need about maybe 2 weeks, 3 weeks before the action becomes efficient. It takes time for the adaptation to happen. Well, if you do this in the young animals, you have actually the opposite effect, and this has been observed. So if fluoxetine is administered during the first 2 postnatal weeks of the life of a mouse, and you look at the phenotype later, you actually see an increased anxiety and depression-like behavior. So this is very strange and very unusual. So what is happening in the brain at that time? This is something that we wanted to look. So is this anything to do with what we saw in the barrel cortex and the sensory cortex? We thought that this maybe we need to look at other circuits, brain circuits. So we turned to the expression maybe in the prefrontal cortex. And in the prefrontal cortex, what we had seen is that during the development, during development, there's a transient expression of the serotonin transporter in the prefrontal cortex that you can see here, with in situ hybridization in the deep layers of the cortex. And using a fate mapping of SERT-CRE, you could see, so I don't know how familiar you are with this, but here is a transgenic mouse where the CRE recombinase has been inserted inside of the SERT gene. And so if you cross it then with a GFP reporter, you're going to be able to see all the neurons that have expressed the serotonin transporter at one time or another during development. So this gives you a very nice mapping of all the brain areas that express the serotonin transporter during development. And we could see this is very broad. Actually, it concerns, of course, the raphe, which is the main area in adults. But during development, it concerns also the thalamus, the prefrontal cortex. So we focused on the prefrontal cortex. And what we did then was to use these mice where we could label these neurons with a fate mapping and have GFP expressed in them. And then so these neurons are neurons that we could sort, fax them, and then do RNA sequencing to look at the genes that are expressed in these neurons. So we could see that these are not, they don't have any TPH, they don't have any other molecules that make them catecholaminergic or serotoninergic. And they're real glutamatergic neurons, which are, whose phenotype is that of layer 5 and layer 6. And this is, and I don't want to bother you about this, but these are the genes, the type of genes that allow us to identify these neurons as layer 5 or layer 6. And you can see this nice labeling here of these in green of these neurons in layer 6. So what is serotonin transporting doing in glutamatergic neurons in the cortex at the time? It's very strange. They don't produce serotonin. Why do they have the transporter? The transporter is expressed only from, if you look at here, these in situ hybridization from P4 to P10, and then it stops. And what we think, based on pharmacological experiments and histology, is that this is probably a system where the serotonin, which is in excess, gets in the cell and is degraded. And it's a way of clearance and reducing the level of serotonin transiently. So if you block it, you're going to cause maybe some problems. So these neurons are layer 6 neurons. We could see with some injecting some tracers selectively in these neurons using, again, this very precious mouse model where the Cree has been inserted in the SERT gene. And so when we inject a virus, it's going to recombine only in these neurons, okay? And then you can trace the connections and see where these neurons are projecting. So they project mainly to the thalamus and to the brainstem and to the raphe nucleus in particular. So so this is the summary of these projection of these glutamatergic neurons. And we decided to concentrate on one pathway, which is the pathway that goes from the prefrontal cortex back to the raphe, and which has been implicated in the literature to have a very role in the effect of modulating stress responses and modifying the response to social defeat. Also, it's been shown, even in humans, that if you stimulate this region, you have an antidepressant, this medial forebrain prefrontal cortex, if it's stimulated with deep brain stimulation, is one of the regions which causes an antidepressant effect. So when we looked at this, so the way we did this, so we used these two models, either the knockout or to increase the serotonin, either constitutive knockout or just a treatment with fluoxetine during postnatal life. And then we did, so this was done during the critical period from P2 to P14, and then we did some analysis of the morphology and slice physiology when the mice were grown, and we did some behavioral studies and in vivo physiology in adults. So just to show you what we did in terms of morphology, this is a very nice method that one of my postdocs, Mariano Souza-Rey, imported to the lab, and that it's a method which is intermediate between microscopy and electron microscopy, and allows you to have a very fine analysis of what's happening at the synaptic scale. The way you do this is to do thin slices, serial thin slices, and you can look then at a number of different markers which can mark the synapse and can mark the identity, the molecular identity of the neurons. So then when you do the stack, you can really measure the number of synapses going to a defined region. And because the afferents, the prefrontal afferents that arrive to the raphe are only labeled with a transporter called the VGLUT1, we could really define that these were, the picture looks like this, not very dramatic, but the nice thing is that it allows very serious quantifications of these synaptic boutons. And what Mariana could see was that there was an increase of these synaptic boutons on the serotonin raphe neurons. And this was selective for the VGLUT1, so meaning the cortical afferents, rather than the other glutamatergic or GABA afferents. So there was a selective hyperinnervation of these neurons. And this was confirmed with some electrophysiological slice preparation done with a colleague, Manuel Mameli, where we did some injections of channel rhodopsin in these serotonin transporter expressing neurons of the prefrontal cortex. And so the channel rhodopsin, I don't know how familiar you are, but this allows you then to, with a laser, to stimulate these neurons and induce some release. So what we could see here in the serotonin neurons is that in the knockout or in fluoxetine-treated mice during postnatal life, there was an increase in these synaptic glutamatergic influence, also corroborating the morphological analysis. What happens in the prefrontal cortex is a bit different. So there is actually a decrease in the global activity of the neurons, which can be seen with the analysis of frequency of oscillations and the average normalized power that you can see in the cortex. If you analyze this by frequency and the average normalized power, you can see that in controls, this is the normal curve. And in the mice that have been treated by fluoxetine during this early postnatal period, there is a decrease in this overall power spectrum, so meaning that there is this decreased activity. And this was verified with local field potentials and signal unit recordings in the PFC of adults, even in behaving animals. And at the single cell level, there was some recordings that could show very well that in these neurons which had expressed transiently the serotonin transporter, there was a decrease in the frequency, the spike frequency of these neurons when these mice had been treated with fluoxetine during early life. So what about the behavior? So behavior, we did a few tests that had been almost published before, but we replicated this. And one of the tests that is currently used to assess depression is the novelty suppressed feeding. I don't know how much familiar you are with this, but so the mouse is not eating for 24 hours, then you put him in an open space with some food, and the mouse is normally afraid to go in an open space, but he's of course hungry. So you measure the time that he goes to the food, and this gives you a sort of measure, because when they are very anxious, they take a longer time to go to the food. And the other test, which can be criticized, but which is the forced swim test, where the mouse is put in warm water, and you count how much time he or she is paddling around and swimming. So these are standard measures. I mean, you can criticize them, but they're standard measures for assessing depression in rodents. And in what you can see here, these are individual cases, for the latency to feed is increased in the mice that have been treated with fluoxetine during postnatal life, and they have an increased time of immobility in the forced swim test. So are these now the question? Okay, we see some behavior, we see some increase in synaptic activity in the raphe. We see this both with the morphology and with physiology. We see this behavior, but are these connected? It could be something completely irrelevant. So to question this, what we did was to try to insert a dread in these neurons selectively. So the way we did this was, again, using this genetic mouse model. We inserted an AAV, a virus, which will express conditionally this HMD4 gene, which is a dread which will be selectively activating after CNO administration. And so this, if you do this during the time when CERT is expressed, you're going to have the expression only in these neurons which express the serotonin transporter. And then you can test them in adults and see what happens. So the pharmacogenetic manipulation, so these receptors have the particularity of being activated when you inject a molecule which is called CNO, and which is going to activate selectively the neurons which are expressing this GQ dread. And what you can see here is that so here is the same experiment I showed you previously. So in the fluoxetine-treated mice, there's an increase in latency to feed. And if you, at the same time, do a stimulation with CNO to activate the neurons in the prefrontal cortex, you actually save the behavior, so they now have a normal latency to feed. Same is observed in for the forced swim test. So the immobility here is reduced, recovered to normal in the after cyano treatment. So if I just maybe recapitulate what I showed you in these experiments, so what we have here is a situation when if the animal is treated by fluoxetine, which increases the level of serotonin during the first 2 postnatal weeks, the consequence on the prefrontal cortex will be a decrease in global activity here. And in terms of anatomy, there was actually an increase in the innervation and in synaptic drive, as if there was some compensation to compensate for the fact that the cortex is now more silent. This is just an explanation. Okay, so I don't know where I am here, but yeah. So the next question was, okay, which receptor is involved? Is this again the 5-HT1B receptor as we saw in the thalamic axons? But now we have this modern way of looking at the neurons, and so we can look at single cell and expression on the transcriptome, and that's the beauty of the transcriptome, I must say, is that you can, with one single experiment, look at thousands of genes and see which one of your interest is changed or not. And so in these experiments where we had extracted these neurons and then done the transcriptome, we could look actually which were the receptors that are expressed in these neurons. And here is a graph showing all these receptors, all the 5-HT receptors. And as you can see, the 5-HT1B is not a good candidate because it's not expressed. And what was striking was this receptor, 5-HT7, which I had never heard about before. But then we started working a bit on this receptor. We showed that actually it is expressed. We can see this expression in the prefrontal cortex at the same time as the serotonin transporter is expressed. This is a double in situ hybridization where you can see in green the serotonin transporter with these green dots, and the 5-HT7 receptor with these red dots. These are the mRNA, of course, not the protein. So is this receptor really important? Again, we used different methods, genetic, 5-HT7 knockout mice that were given to us by some colleagues in Rouen, and overexpression of the 5-HT7 receptor with an AAV that's been given to us by Evgeny Ponymaskin, and some pharmacology, because interestingly, there are very good agonists and antagonists for this receptor, very specific. So I will not go into the detail of this, but just that we did the same experiment that I showed you previously. So treating the mice either with sucrose or fluoxetine from P2 to P14, same protocol, but this time we did this in the controls and in the knockout. And then we did the behavioral testing. And here, again, novelty suppressed feeding is normalized in the knockout. Same for the immobility time in the forced swim test. This is normalized again. So apparently this behavior here is really 5-HT7 dependent. Converse experiments, what we did here was inject a virus that expresses the 5-HT7 receptor in excess. So this time it's an overexpression of the 5-HT7. And this time we could actually, just by doing this overexpression, have a sort of anxiety-like depression in this model. So, so just to summarize, I'm sorry. Yeah, here, so during development, what we can say now is that in these neurons in the prefrontal cortex, 5-HT7, the serotonin transporter and the 5-HT7 receptor are present in the same neurons. These are neurons that go to different places, thalamus, but also in the raphe. And in the normal control, when the serotonin level is good, you have a normal level of activity. And the controlled emotional responses. But when you increase the level of 5-HT7, or when you decrease the serotonin transporter, you have a phenotype which results in this hyperinnervation, reduced activity in the cortex, and inadequate response to stress in like a feeding test or the immobility in the forced swim test. Okay, so we've done a lot of knockouts and so on. And for the moment, I only told you about, what happens when serotonin is increased. And I showed you that there's an effect in the sensory circuits. I didn't mention all the circuits that we analyzed. We had analyzed also the visual and the auditory system, etc., and found similar things. But okay, so this is all an increase. And also the question is, what happens when you decrease serotonin? Yeah, that's a sort of obvious question. And actually, when I think retrospectively, I was lucky not to start with this because we found actually very little on the brain development. So let me show you what we studied as models. We studied actually 3 or 4 different models of serotonin depletion. One of them was the developmental model. I'm not going to go into the details of it, but it's a model where a transcription factor, which is required for the development of the serotonin neurons, is lost. It's called PET1. It's a transcription factor. So when it's lost, serotonin neurons don't develop. So that's a hyposerotonin model. Another model is when you reduce the vesicular monoamine transporter. When the serotonin is not stored in the vesicle, it's completely degraded, serotonin goes down. And finally, the other model, which is maybe the more pure, is a TPH2 knockout, where you selectively remove TPH2 in the genome. And in this case, you can still have the TPH1 in the periphery, in the blood, in the gut, and so on, but in the brain, you're not producing serotonin. Okay, the phenotypes are more or less similar in these mice. So what was I think common in all these models was mainly in the TPH2 knockouts and the vesicular monoamine transporter knockouts was a severe growth retardation. So you can see here, in terms of this is the age in days, and this is the weight. And so this would be a normal developmental curve for a mouse. And you can see here in the knockout that this curve is very, very different after P7. It goes down. And if you look at mice here, age P10, you can clearly identify which is the knockout because they're half the size. But eventually, if they survive, they catch up and they're pretty well in terms of weight. So we looked at all our favorite areas and looked at the barrel cortex. We looked at this prefrontal cortex and many areas. We didn't find any of the changes that I mentioned previously. But the only thing that we found was that there's sort of a delay in the maturation of the cortex, which you can see here at P7. So this is a normal cortex in a mouse. And this is a blow-up of you can see here the upper layers, layer 4, layer 6 here. And here you can see in a hyposerotonin mouse that the upper layers here there's a blow-up, are much thinner than in the control. So you can see this very clearly at P7, so 1 week after birth. But eventually, if you look 1-2 weeks later, it normalizes and it catches up. So we didn't see many morphological details. We haven't pushed, maybe if one looks in the great detail, one will find something, but we didn't find anything very, very obvious. So the idea for me is that if you're reducing serotonin from early life, you have other mechanisms that are compensating in this system. There are other, nevertheless, many behavioral alterations in these mice. Curiously, one of the most dramatic is the increase in male aggression. That's been found in all the models. The mice which have lower serotonin are increasingly aggressive. And there is a deficit in the maternal care, meaning that the mothers which are hyposerotoninergic, don't care well for their pups, and the pups die. In terms of anxiety, curiously, it's rather than having an increased anxiety, these mice have a decreased anxiety. They explore more in an open space and in elevated plus maze, but they have an increased learned fear experience. So this is something that we observed. In terms of depression, we didn't see any increase in depression-like phenotypes. And neither did our colleagues, and this has been confirmed by many other colleagues in the field. However, none of these behaviors seems to be developmental because they can all be corrected, they can all be reproduced if you decrease the serotonin level in adults, and they can be rescued if you normalize the serotonin levels. So they don't seem to be developmental, and I'll show you just one example, which is learning, the effect on learning. We found that these mice, the hyposerotonin mice, have a learning deficit. The way we explore this is the novel object recognition. So you put the mouse in a cage, in the home cage, you put two objects, and you look, and they normally explore this. And 2 hours later or 24 hours later, you change one of the objects, and then you measure how much they go to one or the other object. And normally the mice are curious, so they'll go to the new object, and then you make a ratio of the time they spend on the novel object versus the total time, and this gives you a recognition index. So normally, this is with the training time in wild type in blue, and you can see that the recognition index is increased in the wild type mice, and in the knockout, this is very much reduced. So they don't learn even 2 hours later, they already forgot what is the new object. So is this anything developmental? The way we did this was using, again, this dread morphology, and here maybe in this slide it's better explained. These are these receptors that are artificial receptors that you insert into the genome of the mice, and that going to express this G-coupled receptor, which is coupled to GQ and going to have an excitatory effect. And you can measure this here. You can put a tag on it to see that it's well expressed. And you can see here here are two different viruses, one excitatory, HM4+, and you can see here on a slice preparation that if you put cyano on the preparation, the normal firing of the serotonin neuron goes down, so that works pretty well. And if you the inhibitory effect is really working very, very well in this. I think, this is just the control. So the way Sebastian did this experiment was to habituate the animals to do a learning training task, and he injected in the adult mice the dreads either in the dorsal raphe or in the median raphe, and then when he did his test habituation, he injected saline or cyano at different steps of the training. And what he could see actually was that in the when he injected in the dorsal raphe the cyano, so when he was inhibiting the neurons of the dorsal raphe, you had no effect on the recognition index. I'm getting mixed up here. And when you inject it in the median raphe, you have a reduction in the recognition index. So this is showing you that if you inhibit the activity of the median raphe during the training of the test, you are actually preventing the acquisition of this learning. So this clearly showed that this was an adult function of the serotonin neurons. So I'm done now with showing you this. So if I summarize what I showed you the experiment showing the increasing of serotonin. I showed you that this alters several sensory systems, sensory circuits and prefrontal circuits. I showed you that there are some consequences on adult emotional behavior in terms of stress control. And the effects of decreasing serotonin, that there are minor developmental effects, no clear-cut circuit changes, but that adult depletion causes important changes in many innate behaviors, such as learning, impulsivity, maternal behavior, sleep, etc. I didn't show you all these, but because for lack of time, but that's mainly from our experience and that of the experience of others, doesn't seem to be any results of any deficit of serotonin during development. So just the take-home message that I wanted to give you in a more broad term is that why are we acting on serotonin? Actually, because serotonin is just in the center, like many other maybe transmitters or other monoamines, in that it serves as an interpretation of what's happening in the periphery, in the environment, and then translating it into a signal which is happening in the brain and then can have an effect. So this can be an environmental challenge that happens during development. It can be a pharmacological exposure, as we mentioned, exposure to antidepressants, could be pesticides or whatever. And it could be drugs such as, LSD, ketamine, all these drugs which are interfering with the serotonin or other neurotransmitters. And these, if they act at the time when the circuits are starting to be built or are developing, plus if, as we heard also, Dr. Miskolczy was interested in the effect of genetics and environment, if you also have a genetic predisposition, could lead to important circuit changes that could lead then to behavioral disorders, either overt behavioral disorders or just increased vulnerability to any disorders that could happen in later life, and comes back to the idea for schizophrenia, for instance, of a double hit model where you could have genetic predisposition and then some environmental changes that happen. So I'm done now, and just want to acknowledge many generations of students that's been working with me, and they were really fantastic to work with, postdocs. I can't name them all, but they're all in my memory, and they're part of my family. I'm retired now. This is the last, I think I retired in 2018, 19, and this is the last picture before I retired of the group of my team. I continue working a little bit, and I joined actually the teams of younger researchers, first in the Institut du Cerveau et de la Moelle with Nicolas Régnier, but now I'm hosted by this very nice person, Anne Roumier, who's here in this picture, and I'm part of it, and it's part of the Sorbonne University, and we are working on the neuroimmunity, development and plasticity, and actually looking at how now serotonin, I'm no longer working with the neurons, but how they're acting on the immune system and on the microglia, macrophage, and so on. So that's the new direction that we're taking. And so you're welcome to visit.